Okay, so the way we are going to talk about cells, we're gonna break it up into parts. Okay, so let's start by talking about the 3 regions of a cell. Okay, and so I'm showing you this on an animal cell, but it's the same on plant cells or really any eukaryotic cell. Okay, so the 3 sort of like main regions of a cell include the plasma membrane, which is the outer boundary of the cell. So essentially it's the outside. Okay, so that's one. Then the nucleus. So the nucleus, I shouldn't circle it there. I should circle where it says that. Move my, right? That's the second area. Okay, and then the third region of the cell essentially is everything from here. Oh, why did it do that? Gosh, come on, pen. Everything from there to there, right? So all of that, okay? And you know, here, yeah, that would be number 3. And that is collectively called the cytoplasm. So I'm going to erase it just because it's clogging up the diagram. Okay. So number 3 is the can't spell. Cytoplasm. Okay. Now the cytoplasm has several parts as well. The cytoplasm has little thingies floating around in it. And those thingies are called organelles. So that includes organelles. I'm skipping where it says flagellum. I didn't plan this well. Organelles, right? And then the fluid component is called, I'm going to squeeze it in here, cytosol, right? So the cytoplasm includes cytosol as well as the organelles. So the cytosol is like the liquid that's all throughout the inside of the cell. And then the organelles are like suspended within the cytosol. And collectively, that whole thing we call the cytoplasm. Sometimes when people are feeling sloppy, they kind of interchange, they use them interchangeably, cytoplasm, cytosol. Ideally, we wouldn't do that, but you know what? It happens, okay? So this video is going to be focused basically entirely on the plasma membrane, because in addition to talking about the structure of the plasma membrane, we're also going to talk about how materials move through the plasma membrane. Okay, so that's what the rest of this video is going to be about. And then there'll be another video that talks about the nucleus and, you know, sort of the parts of the nucleus, what the nucleus does, and all of the organelles. Okay, so 2 and 3, the final 2 regions. Okay, so that's what we're doing. All right, so first thing I want to say about the plasma membrane is you will notice that it's found in all types of cells. It was a prolonged all, right? So plasma membrane, it's found in prokaryotic cells. Plasma membrane, found in animal cells. Plasma membrane, found in plant cells too, okay? So it's found in everyone, okay? The primary function of the plasma membrane is the sort of external boundary of the cell, as well as regulating what goes in and out of a cell. Okay, so remember that we talked about this idea that cells are the smallest units that have the characteristics of life. And one of those characteristics is having an inside that is distinct from the outside world, right? Having some sort of barrier, okay? All right, so one of the things I love about your book, okay, is that they include these little diagrams that are like black and white, except for the structure they're trying to get you to think about at any given time. So in this one, they're trying to show you, oh, here's where the plasma membrane is found in each of these different cell types, right? So in a plant cell, it's right there. In an animal cell, it's there. In a bacterial cell, it's there. Oh, that's cool. Whatever. Okay. All right, so function of the plasma membrane is boundary. And then additionally, it also allows certain substances to move in or out of cells. Okay, so those are its functions. The plasma membrane is primarily, not completely, but primarily a phospholipid bilayer. So phospholipid bilayer. Okay, so we did talk about phospholipids for like half of a second when we were talking about lipids. We're going to talk more about them now. Okay, they are a type of lipid, as the name would imply, and phospholipids have an interesting structure in that they have these two long fatty acid tails, right? So that's this part down here, these. And those fatty acid tails always face the interior portion of the bilayer. So notice when you look at this, right? Notice that there's one layer of phospholipids here, and then there's another layer of phospholipids here, hence the name phospholipid bilayer, okay? The reason that those tails point towards each other is that the tails are all what we call hydrophobic. This is not the first time we've heard this term, right? Remember, they're fatty, yeah? So hydrophobic, what does that mean? Yes, the literal translation means water-fearing, but in reality, it means that it's repelled by water, okay? So an important thing to realize about cells is that the exterior environment of a cell is watery. The interior environment of the cell, the cytoplasm, is also watery, right? So they're in water, they're filled with water, it's water everywhere, okay? And so those hydrophobic tails are kind of spontaneously forced next to each other because they are so hydrophobic, okay? And the reason that they're hydrophobic is because they are nonpolar. That's another term that you already know, okay? Now, the head of the phospholipid is hydrophilic. I believe this is the first time we've used this term, but it doesn't matter because you can figure it out. It's the opposite of hydrophobic. So philia, literally the Greek root philia means like sisterly love or fondness. Okay, so hydrophilic literally means water loving. Once again, though, molecules don't have emotions. So it just means attracted to water. Yeah. Okay, so that the head, the phosphate head on the phospholipid is hydrophilic, and that's why it's on the outsides of the bilayer. So the outer surface, you know, the extracellular environment, extracellular means outside of the cell, and then the intracellular environment, the cytoplasm, which are, remember, both of those places are super watery. Okay, so that's the basic like setup of the phospholipid bilayer. And phospholipids will do this spontaneously. So if you had, you know, a handful of phospholipids and you chucked them in some water, they would do this spontaneously because of the hydrophobic nature of the tails. They just automatically orient themselves so that the tails are next to other tails. Okay, spontaneous. All right. Excuse me. Whew. Sorry, I had like a little, I didn't know if I was going to sneeze or what, but anyway, sometimes people talk about a plasma membrane, they describe it as a fluid mosaic model. This membrane is actually quite flexible, okay? And in addition to the phospholipids that make up the bulk of the membrane, there's also other things kind of peppered throughout, right? So there are a lot of proteins that are found kind of throughout the membrane, and those proteins have a ton of different functions, right? So, oh look, hydrophobic, hydrophilic, there's the bilayer, there's the extracellular fluid and the cytoplasm. Anyway, okay. So these kind of purpley magenta, whatever, are representing proteins, okay? And so proteins are sort of scattered throughout. These little green things represent carbohydrates that are generally used for like identification. They're kind of like little flags that are like, you know, the way that cells can kind of identify each other, okay? One of the things that I think that your book leaves out that's kind of unfortunate is it doesn't really address the incredible diversity of those membrane proteins. So membrane proteins can have all kinds of functions. They can act as little channels, right? So like in this example, transporting things inside or outside of a cell. They can be enzymes. So we talked about what enzymes do in the last chapter, right? So they can be enzymes that are bound to the membrane. They can be used for recognition. They can be used as little receptors for communication within the cell, right? And so once again, we're seeing all of these different, right, functions, right, of the proteins. So there's proteins all throughout the membrane. Yeah. And so I don't understand why they keep switching color. It's like they're purple here and then they're like magenta here. I don't know, but whatever. Okay. So that's what they do, all kinds of things. Okay. One of the ways that I like to have students kind of think about that fluid mosaic is to do a little thought exercise. Okay. So imagine you get yourself like one of those plastic kiddie pools, like a baby pool. Yeah. Okay. Fill it up with water, put in a ton of ping pong balls, right? And so they're all going to be floating on the surface. Yeah. All the ping pong balls. And then what I want you to do is I want you to throw in a bunch of other balls. Okay. So, you know, throw in a volleyball and a basketball and a softball and a golf ball. Oh, I don't care, whatever. In your imagination, throw in some balls. They're all going to float to, not a bowling ball because it doesn't float. Pick balls that float in your imagination. Okay. And so if you stand and you look down at that baby pool, right? The ping pong balls kind of represent like the surface, right? The phosphate heads, right, that you can see on the surface. And then all the other balls, the, you know, soccer ball and basketball and volleyball and whatever, those represent the different proteins, okay? And so they call it a fluid mosaic model because if you like blew on it or you like nudged one of the little balls, they'd kind of, you know, move around in the little pool, okay? Now, that can be sort of problematic actually, because you don't want it to be too flexible because then it'll just kind of like fall apart, right? Or proteins will move to places they shouldn't be. Yeah. And so in addition to all of those proteins and all of those phospholipids, you also have little cholesterol molecules. And so the way that, what do I want to say? So, you know, this picture is from a different book because yours doesn't show any cholesterol, any of your pictures. None of these show any cholesterol molecules, right? That one doesn't either. Okay. So yeah. So I'm using a picture from a different book just to kind of, so you can see the relative size. So the little blue and green things are supposed to be the phospholipids, and then the little yellow guys are cholesterols. And so the cholesterol, essentially what it does is, so it says helps membrane retain its flexibility. Yes, but it also kind of gives it some structure. So go back to your baby pool, your imaginary baby pool. And now let's take like a volleyball net or like the net from a soccer goal. Okay. And like place it over the top, right? Well, everything's still going to be able to move a little bit, but things aren't going to be able to move as much. That's kind of what the cholesterol does. So it kind of keeps everything together. Okay. Right. So notice that when we look at images like this, the plasma membrane is just this thin thing and you can't see a bilayer because the bilayer is super duper small, right? It's super thin. So at this level where you're looking at an entire cell, you can't see the bilayer just because it's you know, so small. Okay, so you never see it in an image like this. When you look at a image of an entire cell, whether it's a bacterial cell, or whether it's a plant cell or an animal cell, it doesn't matter. They always, when you're looking at the whole cell, they show the plasma membrane as if it looks like a sheet, but in reality, right, when you zoom in far enough, it looks like that bilayer. Okay, so anyway, all right. So all those proteins that do all kinds of cool things and whatever. All right. So as promised, the other part of this that we need to talk about is we need to talk about how substances move through the membrane. Okay. So there's two sort of major ways that materials move in and out of the cell. One type of membrane transport is called passive transport. And what that means is that the cell does not need to expend any energy in order to move something. Cell does not expend energy, right? So it doesn't cost the cell anything in order to do that, okay? There are three main types of passive transport. We're going to focus on two of them, okay? And we're going to talk about them a little bit more than your book does. Not a ton, but a bit. Mostly because I really like to talk about osmosis, because osmosis is a useful life skill. It is a useful life skill. And so today, I'm not going to tell you the results, but today I did the lab, the second part of the lab that you guys are doing this week. I have done the first part at home before, so I didn't bother doing that again. But the second part of the lab that you guys are doing this week, I hadn't done at home before until today. And it worked perfectly, by the way, so it should work fine for you all, okay? Unlike some of our other interesting little surprises that have happened with other labs. Anyway, what do I want to say about that? Yes. Okay. So I did that little second part of the lab at home. And afterwards I kind of quizzed my husband. I was like, so if I tell you, there's these veggie slices that I put in these 3 different solutions, what do you think which one was in which? He had no idea, because he doesn't know about osmosis. And I was like, how do you not know about this? This is really important. Anyway, so this is a life skill, and I'll explain to you why in a minute. Okay, so let's start with diffusion first, okay? So diffusion is something that you all are familiar with, whether you realize it or not, okay? So essentially what diffusion is a spontaneous process. Spontaneous. And you know what I mean when I say spontaneous, right? Like it just happens. Okay, so it's a spontaneous process where substances called solutes. So in a previous video, we talked about the terms solute, solvent, and solution when we were talking about water. So remember that a solute is the substance that is dissolved into a solvent, which is usually water, when, for our purposes. And then together we refer to that whole thing as a solution, okay? So solutes are dissolved in water, essentially, okay? So diffusion, simple diffusion, is a spontaneous process where solutes move from areas of high concentration to low concentration. Running out of space, okay? You're familiar with this if you've ever made a solution, right? So for example, if you've ever made Kool-Aid or any kind of powdered drink type of beverage, okay? You put the powder in the water. If you don't shake it right away, at first, all the powders kind of clump together. but then gradually it kind of spreads evenly throughout the water, right? And it kind of spreads out completely. That's diffusion, right? That's what you're observing is diffusion, okay? And so there are some things that can easily diffuse across a membrane, right? So respiratory gases like oxygen and carbon dioxide can easily just go right across through that phospholipid bilayer because the molecules are small and because the molecules are not strongly polar, okay? Anything that is very polar is going to have a hard time getting through those tails. So things that are polar can't just go through, okay? Things that are polar have to travel through little channels. And so they undergo facilitated diffusion. Facilitated just means it's like assisted, right? So if you're doing diffusion directly through the membrane, we call that diffusion or simple diffusion. If they're going through the membrane and they're going from an area of high concentration to low concentration, but they are going through a channel because they can't go directly through the bilayer, we call it facilitated diffusion. Okay. All right, where were we? Where were we? Okay, so this example is showing you diffusion through a semi-permeable membrane. So I didn't even use this word yet, which is unfortunate. Okay, so another sort of defining feature or word that we use to describe a plasma membrane is the plasma membrane is a selectively permeable membrane. That's something that we call it. Okay, because some things can go in and out, other things cannot. Selectively permeable just means things can go in and out. Okay, I'm totally loving the fact that now my computer will let me do backgrounds. Do you guys know where I am right now? No? I'll let you figure it out. Okay, so in this example, they're making up just sort of a generic, okay, let's say we have these molecules of dye, and this is a semi-permeable membrane, not necessarily a selectively permeable membrane, but it's permeable to some things and not other things. Okay, and so what they're trying to illustrate here is, let's say that we had this membrane and there's water on either side, and we put the dye on one side. Well, the dye molecules spontaneously move around, right? And over time, they gradually will go through these little openings, right? And some of them will also go back and forth, right? But overall, the net movement is going to be from, in this example, from left to right, from an area where they're highly concentrated to an area where they're less concentrated, right? Essentially just because they bump into each other and they spread out. Eventually, they will come to equilibrium, which means that the number that are going right and the number that are going left are going to be roughly equal, and they're still moving around, right? It's not like they get where they're going and then they stop, right? They're still moving around, but they're not, but there's no concentration difference on either side, okay? So what else do I want to say about this? So why are these molecules moving in the first place? All molecules vibrate. Essentially, that's what temperature is, by the way. Temperature is a measurement of energy, and is all about how quickly molecules are moving, right? And so the warmer a liquid is, the more the molecules inside of that liquid are moving around, okay? And that's also just, you know, spontaneous. It's called Brownian movement if you want to know. It's physics, whatever, but we're not going to go there, okay? So they spread out. All right, here in this little example, right, and I'm using pictures from other books because your book doesn't do a great job of explaining this, I don't think. Here now they're showing you, okay, let's say we put in that membrane, and on one side we got blue and on one side we got red, and so, you know, the blue is going to move to the right, the red is going to move to the left, eventually they're both going to come to equilibrium, okay? All right. Diffusion and osmosis. But diffusion is one of the things that you are going to be observing in lab this week. So we're using a special kind of tubing called dialysis tubing. And it, as you might expect based on its name, it's used in the process of dialysis, okay? And essentially what it is, it's a semi-permeable membrane that's in tube form. And in the way it's stored is it's dry, and it is like crinkly. It feels like crinkly plastic, basically. As soon as you get it wet, though, it gets all squishy, okay? And it's a tube. And so when it's stored flat, and then you kind of like rub it and open it up once it's wet, and it's a tube, okay? And so what we're going to do is we're going to put some stuff in the tube, and then we're going to close up the tube, and then we're going to stick the tube in another, glass of stuff. And we're going to watch things move inside the tube, outside the tube, which things can move inside of the tube, which things cannot, which things can go, both directions. Be very exciting. Okay. So, anyway, all right. So that's diffusion. So your lab is going to, one of your lab activities is going to illustrate something kind of like this, not with dye, right? With other stuff, but anyway. Okay. All right. Osmosis is a specialized type of diffusion where we're talking specifically about water. Okay, so, oh, look, they look like Mickey Mouse. That's not surprising. Okay, so osmosis essentially is a type of facilitated diffusion, but when we're talking about osmosis, we're talking specifically about the movement of water. Okay, so specifically, movement of water. Okay, and so what was our definition for diffusion again? Let's go back to that for a second, because we need to add a little bit to this. Okay, so our definition of diffusion was, it is a spontaneous process where solutes move from areas of high concentration to low concentration. Okay, another way of describing this is we say that in diffusion, solutes move down the concentration gradient. That's just like a short way of saying going from high to low. Okay. All right. So when we're talking about osmosis, we're talking about water diffusing. So we're talking about water going from areas where there's lots of water to areas where there's less water. Okay. And so there's vocabulary here, and it's kind of crazy. Okay. And so this is the second part of the lab that you guys are going to be, sorry, I just want to see what pictures I had in here just to remind myself. Okay, so this is the second part. This is the second part of the lab, is you're going to be sort of exploring osmosis. Okay, so what you're looking at in this picture is you are looking at what is called a U-tube for obvious reasons, not YouTube. U-tube. Okay, and at the bottom of the tube, we have this selectively permeable membrane. Boop, boop, boop. And so selectively permeable because water can go through it, right? But the sugar molecules, the solute in this particular solution, is large enough molecule that it can't cram its way through that little space. Okay, so we have set up this U-tube with two different sugar solutions. Okay, so on one side, we have a smaller quantity of solute or sugar. Okay, on the other side, right, so there's only 1, 2, 3, 4, 5, 6 sugar molecules. Okay, on this side, it's a much higher concentration of that solute. Okay, so the terms that we use to describe these two conditions is we say that a solution can be hypotonic or it can be hypertonic. So let's think about the roots of those words for a second. Okay, so hypo, what does hypo mean? So a hypodermic needle is a needle that pokes below your skin. So hypo means below or less, okay? Hyper means what? What happens when your kids eat too much Halloween candy? They become hyperactive, right? So more active than normal, okay? So hypo means less, hyper means more, right? So we are going to describe the solution on this side as being a hypertonic solution because it has a greater concentration of solute. The solution on this side is a hypotonic solution because it has a smaller concentration of solute. This is the place where I would ask you all if this is okay and you understand this and you're good with the vocabulary and it makes sense. And hopefully you all would say, yes, that's very clear, crystal clear. This is why we have Zoom, so we can talk about things again if you need to, okay? All right, so if we set up this tube like this, right, in the beginning, what's gonna happen is osmosis. Osmosis is gonna happen. So it's not diffusion because remember that in this example, those little sugar molecules, the solutes, can't move through the membrane. So it can't come to equilibrium by the sugar moving because it can't fit through those openings. The only thing that can fit through those openings is the water. So therefore it's osmosis instead of diffusion, okay? And so if you were to go and count and say, remember on this side we had 1, 2, 3, 4, 5, 6 sugar molecules. We still have 1, 2, 3, 4, 5, 6 sugar molecules over here. They have not moved. They are trapped. Okay. And then whatever the other number is on the other side. Okay. But what do we see happening to the water? Which way has the water moved? Right. Well, once again, there's this tendency to come to equilibrium where the concentrations are equal on both sides. Right. That spontaneous tendency. So what's happening is the water has moved that way. And the reason that the water has moved that way is because the solutes can't move, the water has to, okay? And so because this solution is hypotonic, remember hypotonic means less solute. Conversely, that also ends up meaning, relatively speaking, more water. Right? And if something has a higher level of solute, that conversely means it has less water, right? So the water moves from an area where there's more water to an area where there's less water. Does that make sense? I hope it does. I hope that makes sense, right? Okay, because remember, spontaneously, things move from areas of high concentration to low concentration. So in this case, since the solute cannot move, the water has to move. And so the water moves from where it is more highly concentrated, where there's more water, to where there's less water. So overall, it goes in that direction, okay? Now, the terms hypotonic and hypertonic are relative terms. So the solution is hypotonic when comparing it to the other solution, right? The other solution is hypertonic when compared to that first solution, right? When you have solutions that are equal concentration, we say they are isotonic because iso means the same, right? So same concentration of solutes on both sides, which means that a bunch of water had to move over here in order to get to that same concentration. Okay. Yeah. I hope that makes sense. Right. And it actually looks really cool if you do this in real life with a YouTube. They break easily and they're expensive, so we don't usually do it this way for those reasons, because they break and all of that. But plus putting the membrane in and the whole. Anyway, it looks really cool though, because it's like you start off with, because you can't see the sugar when it's dissolved in the water, right? So you start off with two clear solutions on either side of the tube. They don't look any different from each other, right? And then over time, the one side gets lower and lower and lower, and the other side gets higher and higher. And it's like, why is that? Why doesn't gravity just, keep it level? Because of this, right? Because of osmosis. Okay, so we're not going to look at the tube. All right, well, we are going to talk about those. We're going to talk about cells for a minute. Okay, so I'm going to give us a little blank screen to work with here. Okay, so let's, I almost started singing a Salt and Pepper song. If you know what I'm talking about, you either appreciate the classics or you're my age, one or the other. Okay, so let's say, oh, what, dude? Sorry, I was all excited too. All right, here's a little beaker, right? There's a little beaker. Okay, and there's some liquid in it, and here's a cell. Okay, now most cells, I shouldn't say most cells, all cells. Cells are not, they're not filled with pure water, right? They have things dissolved in them, right? So there's a certain solute concentration that is typically found in cells, okay? And so if we're talking about, what am I trying to say? If we're talking about, I don't even know off the top of my head what a typical sugar concentration would be inside of a cell. But if we're talking about salt, say, right, a typical cell in a living thing has a salt concentration of about 0.9% salt. Who knew? Okay. So let's just use that as our example. Okay. So inside of my cell, I have 0.9% NaCl, salt. Okay. Let's say that we tossed our cell in a beaker filled with pure water. Okay. All right. Sweet. Okay. So if it's pure water, that means it's 100% H2O. Okay. What percent of water is the inside of the cell? Well, if it's 0.9% salt, it's going to be 99.1% water, because 100%, just, you know, pretend salt is the only thing in there. Okay. And so since we're talking about osmosis, we're assuming that the salt's not moving and it's only the water that's moving. Okay. So just, you know, pretend with me that that's what's happening. Okay. So which way is the water going to move? Right? If the solution surrounding the cell is 100% water and the solution inside of the cell is 99.1% water, which way is the water going to move? Well, it always moves from where there's more to where there's less, when there's higher concentration to lower concentration. So that means the water is going to go in which direction? It's going to go into the cell. Huh. Okay. So when you put a cell in a solution that is hypotonic, so this is a cell in a hypotonic solution, that's an abbreviation for solution, right? What that means is that the water that's in that solution is going to go into the cell. What is that going to do to the cell? I'm just going to let you think about that for a minute. Okay. All right. Now let's put the cell in a hypertonic solution, because why the heck not? Okay. So here's my, wow, I cannot draw. There's my beaker with my fluid. Here's my cell. Okay. So, and remember, it's the same cell, so it's still 0.9% sodium chloride. Okay. A hypertonic solution would have to have more solute, right? By definition, hypertonic means more solute. So let's say, just let's be crazy and say this is 20% salt, right? So it's a super concentrated salt solution. I don't even know if you can get that very easily. That would be super concentrated. Okay, so whatever. Okay, so 20% salt in the solution. Yeah, only 0.9% inside of the cell. All right, so remember we're talking about osmosis, so we have to figure out how much water is in those two things as well. So if the solution, we already know inside of the cell, it's going to be 99.1% water because we did that math already. Okay, what's the percentage of water in the solution? Well, if it's 20% salt, we're going to assume then that it's 80% water. So now, which way is the water going to be moving? Well, there's 99.1% water inside of the cell. And the only reason I'm writing it out here is just because I can't fit it in there. Okay. But yeah, that's where it is. Okay. So it's 99.1% water on the inside of the cell. It's 80% water outside of the cell. So which way is the water going to spontaneously go? It's going to leave the cell. Huh. All right. Now, cell in isotonic means it's at equilibrium, so nothing happens. Right? So there's no net movement. Okay? All right? Okay. Let's look at some real life examples, shall we? Okay. So this is my favorite. Hold on. I got to get back to here. Okay. So here we have an animal cell and a plant cell. And we have put them into these three different types of solutions to see what's going to happen. All right. So one of the big differences that some of you guys already know because you've learned about this through the process of doing Organism of the Week, you've learned about this. And that is that plant cells, in addition to their plasma membrane, because they do have a plasma membrane, they also have a rigid cell wall around the outside of that. So the cell wall does not play the same role as the plasma membrane, right? Remember, the plasma membrane is that selectively permeable membrane. But the cell wall, wow, I got some height there. Let's just calm that down so that's not too distracting. So I'm distracting myself. That's bad. All right. So the cell wall is for structural support. It doesn't regulate stuff going in and out really at all, right? It's just structural support. It's like scaffolding on the outside of the cell, right? It's not, it doesn't control things moving in or out. Okay. That's just the plasma membrane that does that. Okay. So we got an animal cell, we got a plant cell. Okay. The difference being plant cells have a cell wall, animal cells do not. Okay. So what happens if you put an animal cell in a hypertonic solution? So those little circles are supposed to represent our salt or sugar or whatever the solute is. Okay. So if there's way more of the solute outside of the cell than inside of the cell, then remember water is going to leave the cell. So in animal cells, if they're in a hypertonic solution, they literally shrivel up. So if you were to take some red blood cells, a sample of your blood, and you were to put it in a salty solution, you could watch under a microscope as your red blood cells, which are normally this shape, you could watch them like, right? Because the water is leaving them, so they shrink. Okay? Like a water balloon that has a slow leak in it, they shrink. Okay? All right. What happens if you put an animal cell in a hypotonic solution? So if you go back, rewind a couple seconds, right? What happens in a hypotonic solution is water rushes into the cell. Okay, so if we're talking about an animal cell, if water's rushing into the cell, eventually that plasma membrane is not particularly strong. So eventually that cell is going to burst. It's going to pop like a water balloon that you're trying to fill too much, overfilled. Okay, because it's too much water is going into it and it's just not strong enough to support that. Okay, so it'll pop. Once again, you can do this in the lab, right? If you were to take a sample of your blood, put it on a slide, right? Add some pure water to it. That will be hypotonic to your cells. right? And you could actually watch the cells swell and eventually explode, right? Because the water rushes into the cells. Okay, cool. All right. Animal cells, like our red blood cell in this picture, is very happy in an isotonic solution. Because a little water goes in, a little water goes out. At that equilibrium, it doesn't shrink, it doesn't explode, it's happy, right? So I'm going to put it's not really happy, it doesn't have emotions, but you know, you get the idea. Smiley face. Okay, so that's ideal for an animal cell. Plant cells, though, it's a little different because of that cell wall. Okay, so in plant cells, all right, when they shrink in a hypertonic solution, what's interesting is it does this really weird, it's called plasmolysis is what this is called, but you don't care about what it's called, it doesn't matter. But basically what happens is see how the plasma membrane is like pulling in from the cell wall because it's shrinking inside of that rigid box. Okay, what this always reminds me of, some of you are old enough for this, some of you are not, is wine that comes in a box. Okay, so this is when you've reached the age where you don't care. Okay, and I just need some wine and I need a lot of it and I need it to be cheap. Okay, so some of you you're born that age, right? Some of you, it's like, I like to drink high quality wine for a while until you give up on life, and that's fine. Anyway, some wine in a box is actually good. Some of it's really bad, but some of it is, decent. So the way that works, right, is you have a cardboard box and it has a little spout, right? And it has a bag inside. And when you buy the box and it's full, that bag is full of wine, right? And as you take wine out, the bag shrinks. The box stays its same shape, right? You can't tell from the outside if you're running out of wine. This is the problem with wine in a box, okay? You can't see. So, you could be dangerously low and not realize it, all right? So you take the wine out and the bag shrinks inside of the box, kind of like this, because water, or wine in my example, right, is leaving the bag, okay? That is not good for plant cells because if they shrink too much, the cell wall can actually break. It would be like if you put something heavy on top of your wine in a box. So let's say you had 2 boxes of wine, one sitting on top of the other. And if they were both full of wine, it would be fine. There would be no problem. But if the bottom box was getting empty and the top box was full and it was really heavy, it might cause the bottom box to collapse because there's nothing like supporting the walls of the box anymore, right? Because the bag of wine has shriveled up. The same can happen in a plant cell, right? So the cell wall can actually break if the cell itself gets too small. And if the cell wall breaks, it can pierce the cells and then they're screwed. Okay, so hypertonic is bad for everybody. It is bad, right? Cells do not do well in a hypertonic solution, ever. Animal, plant, don't matter. It's bad. Some archaeans can do it just fine. I don't know. That's weird, right? But animals and plants, hypertonic is bad. Okay. What happens if you put a plant cell in a hypotonic solution? So remember, in a hypotonic solution, water is moving into the cell. Well, the cool thing about plant cells is that because they have that cell wall, what happens is water goes into the cell, but the cell wall kind of limits it. And it's kind of like, all right, we're going to get nice and plump, but no further. It's limited because the cell wall is there providing structural support. Okay. And so the term that we use to describe that is we say that the cell is turgid, right? Which essentially just means that it's like super firm because it's filled with the max amount of water that's pushing on those cell walls. Okay. So let's go back to water balloons for a second. So remember how I said, like, this is a water balloon with a slow leak, right? This is a water balloon where you put too much water in and it pops. Okay. If your water balloon is made of really good, strong latex, sturdy latex, like a cell wall, and you fill it up nice and full, perfectly full, in fact, right? You know this if you've ever had a good water balloon fight as an adult. As a kid, you don't know. You're just throwing balloons. You don't know what you're doing, right? But when you're an adult, when you're digging in the pile of water balloons to find the one to throw at your frenemy, which one do you grab? You don't grab the floppy one because that's just going to bounce off of them. It's just going to be all soft and wobbly and it's just going to bounce, right? You grab the ones that are full because they're firm, they're turgid, and they're going to pop when you throw them at your frenemy. Yes? Sometimes we have water balloon fights in the family at my house. It's can get a little violent. Yeah. This summer, my husband was like, everybody's ganging up on Daddy too much. I'm done with this. He was getting too many turgid water balloons thrown at him, and he didn't like it very much. I think the kids might be too old now. Now they can actually throw. It's bad for Daddy. So they don't throw them at Mom because Mom's like, you can't hit a girl. What are you doing? Yeah, I take advantage of that. Anyway, okay. Right? So plant cells are turgid. They're nice and firm when they're in a hypotonic solution, right? So our plant cell is happy when it's turgid. Now, a plant cell in an isotonic solution is not the end of the world. It's fine, right? Because the amount of water going in and out is fine. It's balanced. But notice that the cell isn't as like firm, okay? And that turgidity, that firmness is really important for plants as a means of supporting the plant. Plants don't have bones or exoskeletons the way that animals do. So their cells provide a lot of the support. Okay, so let's look at this for a second. So here is a happy plant and a wilted plant. The reason that the plant is wilted, the one on the left, is that it is currently, the cells are in a hypertonic solution. So somebody forgot to water that plant. There is not enough water in there. And so the cells are losing water like crazy. The cells are shriveling. And because they're no longer firm and like pushing up against each other, right? It gets wilty. Okay. If your plant is just starting to wilt just a little bit and you catch it and you give it plenty of fresh, clean water, you can reverse that, right? You can bring it back to our turgid state. Okay. But if you're not quick enough, if it's been too hypertonic for too long, those cell walls are damaged and you're not bringing it back. It's dead. You killed your plant. Good job. Yeah. So this plant has nice turgid cells. The cells are full because they're in a hypotonic solution. So the water is going into the cell and it's pushing in there and the cells are pushing on each other because they're all full and turgid. We call that turgor pressure. Go figure. Okay. All right. This is a fun, useful household tip. Okay. Let's say that you have some floppy vegetables in your crisper drawer in your fridge. I don't mean rotten. If it's rotten, you're not resurrecting it. But let's just say, you forgot about the celery, you didn't put it in any plastic, and so your celery is like floppy bendy, right? Or you've got some lettuce that it's not moldy, it's not gotten all slimy and weird, but it's not as crisp and crunch as you want it to be, right? It's gotten a little floppy and sad, okay? Here's what you do. You cut off the ends so it's got a nice fresh cut end, and you stick it in some nice clean water. The best way to do it is put it in distilled water because distilled water doesn't have any solutes in it at all, right? So, but if you don't have distilled water, just tap water is fine, right? Put it in some tap water and guess what? Your celery is going to firm right back up. Okay, this works with carrots too, right? I have a tendency to end up with bendy carrots because they fall out of their plastic bag and then they're sitting in the crisper drawer, they dry out and then they get all bendy and put them in some water. Okay. Conversely, if I put my celery in salt water, super floppy. Okay. You can try this if you have celery at home that has gotten a little floppy. I would make this part of the lab, but I can't be like, okay, everybody, I need you all to get celery 2 weeks in advance and make sure that it doesn't rot, but it's floppy by the time we're having our lab. Right. But if you happen to have any floppy celery, you can try this and share it with everyone if you like. Okay. Anyway. All right. And if you want to make your celery floppy, I don't know why you would, but let's say you wanted to. If you wanted to be able to tie your celery in a knot, put it in some salt water, right? Anyway, okay? I hope it's clear why that would be true. Floppy cells in salt water. Anyway, I almost thought, oh, we're done. Cool. Okay, so all of that, diffusion, osmosis, facilitated diffusion, those are all passive transport. Remember, that means the cell does not expend energy. It's just spontaneous. It just happens, okay? As opposed to active transport. So active transport means the cell must use energy to move something, right? So in this particular example right here, right, this is something called a sodium-potassium pump. You totally don't need to know that, okay? Really common, though, in all kinds of cells in your body. And it's really, really important that you maintain a proper sodium and potassium balance in order for your nervous system to function correctly, and your muscles and a bunch of other cells too, right? They need to, yeah, it's really important, okay? And so essentially what happens in any kind of a solute pump, whether it's a sodium potassium pump or otherwise, is something is being pumped against the concentration gradient. Meaning that it's being pumped from low to high, right? See how the arrow is pointing that way? Okay. And so the only way that can happen, I just went all Minnesota for a second. Oh my. So the only way that can happen is if the cell expends a lot of energy to actively pump something, right? It's the opposite of diffusion, right? So it takes a lot of energy to do, okay? Because the spontaneous thing is the opposite. All right. That's really all you need to know about that. This always requires a protein channel of some kind or another, a protein pump in the membrane. Okay. Another type of active transport is bulk transport. Bulk transport means we're moving something big, which also requires energy, right? Big stuff. So let's say you're not moving like, you know, an ion or sugar molecule. You're moving like a big giant protein, some big solid thing, okay? That's bulk transport. And bulk transport can go inside of the cell or outside of the cell. And those names of those two directions are called endocytosis and exocytosis. So imagine that my hand is the membrane, okay? If some big bulky thing is going to move inside of the cell, what happens is the plasma membrane kind of makes like a dimple, right? And it gets deeper and deeper and deeper until it pinches off. And so this thing right here is just a little pouch of plasma membrane, and we call it a vesicle. So a vesicle is a little like pouch of membrane that is used to transport something around the cell. And so I'm going to zoom in. No, I'm not. Can I? No. Why are you being a jerk? Why are you being like that? Oh, I know, because I'm in presentation mode. It's not going to let me zoom in. You can zoom in, though, on the PowerPoint yourself, right? And so they're trying to show you like, oh, there's the bilayer. You can kind of see it, right? So it's the entire bilayer, right? The entire phospholipid bilayer that gets deeper and deeper and deeper and deeper until it pinches, right? And then now you've got a bundle of stuff inside of a vesicle, okay? All right. So endocytosis, endo means inside, cyte means the cell. So endocytosis is big stuff moving inside of the cell. Exocytosis is big stuff exiting or leaving the cell, yeah? So now we've got a vesicle that contains something, right? And it gets closer and closer and closer to the plasma membrane. Eventually it like sticks to it. And then because it's still the same phospholipid bilayer, it just sticks to it, and then the contents of that little vesicle end up just coming to the outside of the cell. Okay? Exocytosis. Right? So here's another, I like, the reason I included this picture is because I like how it shows you the bilayer a little bit better. Right? So we're looking at exocytosis here. We're looking at endocytosis here. The only difference between them really is the arrows. So if you ignore if you like swap the arrows, they're essentially the same. They're just going in opposite directions. Okay? All right. When a cell is engulfing something that is solid, that particular type of endocytosis is often called phagocytosis. Phago means to eat, right? So I can't remember if that's Latin or Greek, but it means to eat. So phagocytosis is cell eating. And so what you're looking at in this picture, I love this picture because this is an amoeba, which is a type of protist. And this is some kind of algae. It's another kind of protist, some sort of like unicellular green algae probably, okay? And this amoeba is about to eat that thing, right? Well, they're both unicellular. They're both just a single cell. So how does something that is a single cell eat something else? The only way it can, which is endocytosis, right? Amoebas don't have a mouth. So they literally just like engulf into a little vesicle, right? So he's in the process of, he doesn't have a sex either. I'm just, it is in the process of wrapping itself around and engulfing that algae cell, right? And doing phagocytosis. So it's just a specialized type of endocytosis, okay? Anyway. All right, so we'll stop this video here, okay? So that was the plasma membrane, our selectively permeable membrane that acts as the boundary of the cell that lets things in and out selectively. And then we spent quite a bit of time talking about passive transport, 3 different types, and active transport, 2 different sort of main categories, okay? Anyway, all right, so that's the membrane, okay? So in the next video, we're gonna talk about nuclei, and then we're gonna talk about all of the stuff that's in the cytoplasm, all of the organelles, okay? All right, so let's end this one. Stop recording.